Hi everybody, happy Friday the 13th. I hope everybody's been doing well. It is week 7 and today we're going to be focusing on the nervous system and muscles. Alright, so the first part is what would happen if you modified a particular aspect of neuron function. In the following questions, test your understanding of the various parts of the nervous system by asking yourself what would happen if some part was damaged. What would the system still be able to do, and what would it be unable to do? Some nerve gases and insect poisons work by destroying acetylcholine esterase. Acetylcholine esterase is normally present in acetylcholine synapses and acts to degrade acetylcholine. What is likely to happen to nervous transmission in insects exposed to this type of insect poison? So in the CNS of vertebrates, the central nervous system, ACH, acetylcholine, can cause either inhibition or excitation of the postsynaptic neuron, and whether it's inhibitory or excitatory just depends on the type of postsynaptic receptor that is present. So in neuromuscular junctions, which is voluntary muscle, acetylcholine, which I'm going to abbreviate as ACH, is excitatory. So in the heart muscle, the effect of ACH is to reduce the rate and strength of the heart muscle contractions. If acetylcholine esterase is destroyed, then any ACH released into the synapse will then stay in the synapse. And so what this means is if ACH is still in that synapse and it stays there, it's not able to be degraded by the ACH esterase, the postsynaptic neuron will be continuously stimulated by ACH to either depolarize if it's excitatory or hyperpolarize if it's inhibitory. So I'm just going to put remain stimulated by ACH because it could be either excitatory or inhibitory. An example of this is after an action potential is initiated in a muscle fiber, the muscle will contract continuously and the heart rate will slow. The inability to control the contraction and relaxation of muscles like the diaphragm will then result in death. So if that ACH remains in that synapse, then that muscle is going to contract forever, and it can't. There's not enough energy, so that will result in death. And if you're not able to move your diaphragm, if the muscles can't relax and you can't breathe, then you'll die. So, it's pretty important. Happy Friday the 13th. Okay. Number 2. The pufferfish contains the poison tetrodotoxin. I didn't say that out loud before teaching. Some shellfish produce a paralytic poison called saxitoxin. Both of these poisons block the sodium channels in neurons. What specific effects could these toxins have on neuron functions? So if sodium channels were blocked, then no action potentials would generate. Remember when we're talking about depolarization, repolarization, hyperpolarization, that initial action potential is where sodium gates open. And so if the sodium channels and neurons were blocked, then no action potentials would occur. Alright, number 3. A type of spider, the funnel web spider, produces a toxin that blocks calcium channels. A. Can a neuron exposed to this toxin fire an action potential? Explain. They should still be able to fire an action potential because an action potential is more related to sodium and potassium rather than it is calcium. Calcium is more related to what happens to that action potential once it reaches the terminus of that neuron. So just yes for A. It still can fire an action potential. B. Can a neuron transmit a signal across a synapse using neurotransmitters? Explain. So kind of harkening back to a little bit what I said for A, in the synapses, an influx of calcium is what is required to cause those synaptic vesicle. I know that I go between synapse and synapse. I've heard it both ways and I don't know which one, so I'm just going to flip-flop. It's like either and either. So calcium is required to cause those synaptic vesicles to fuse with the membrane of the presynaptic neuron. So if those calcium channels were blocked, no neurotransmitters are released into the synapse. And so the signal can't travel across. So no. Not. Okay, and then to answer your question about how the action potential is more related to sodium than calcium, was when calcium channels are blocked, we said how for B, calcium channels are really only required for the fusion of the synaptic vesicles that are carrying that action potential with the membrane. So the synaptic vesicles are carrying ACH, they're carrying the neurotransmitters. So when calcium is not there to make fusion happen with the membrane of the presynaptic neuron, then those neurotransmitters cannot be released into the synapse, and so the neuron can't transmit a signal. But when talking about action potentials are generated by concentrations of sodium and potassium. So they really have to do with the sodium, potassium pumps that we talk about a lot. And that is what generates the action potential across the neuron. But in terms of transmitting that signal to another neuron, you need calcium to be able to release those neurotransmitters. Does that clear it up a little bit? Maybe? Okay, cool. Yes, of course. All right, let us move on to 4. You isolate a section of a squid giant axon and arrange an experiment so that you can change the solution bathing the axon. You insert an electrode into the axon and place another electrode outside the cell so that you can measure their potential across the cell membrane. With the axon bathed in normal extracellular fluid, you observe a resting potential of negative 70 millivolts and action potentials, when stimulated, that reach positive 55 millivolts. And then here we have a diagram that depicts concentration of each ion in millimoles. And then we have the control normal concentrations in relation to our experimental concentration. We have the ion concentration of sodium and potassium inside and outside the neuron. And we're just going to use this to answer the question below. A. You change the solution bathing the neuron by increasing the potassium concentration to 40 millimoles. What effect will this have on the neuron? For example, will it depolarize the membrane and make it easier to start an action potential? Will it hyperpolarize the membrane and make it more resistant to starting an action potential? Or will it have no effect? Explain your answer. When answering this question, just keep in mind the axon's permeability to potassium, K+, is much greater than to something like chloride, which is a negative ion, which is even greater than its permeability to sodium. At rest, the concentration of negative ions inside and outside the axon are approximately equal, and the equilibrium potential for each ion is dependent on its concentration gradient and the electrical gradient. So we talked about concentration. That is kind of basic. It wants to move from high potential to low potential. That's the gradient. The electrical gradient now has to do with charge. How negative is it? How positive is it? So to answer this, by increasing the K+ concentration to 40 millimoles, and what effects will it have? Potassium ions cannot be added without simultaneously adding counterbalancing anion like chloride. So K+ added without counterbalancing balanced ion. And so as a result, addition of KCl adds both potassium and chloride to the outside of the membrane, which disrupts the the chloride and potassium equilibrium potentials. So addition of KCl disrupts both K plus and Cl minus equilibrium. And because we said earlier, K plus is more permeable, and we said it's bathing the neuron by increasing the K plus concentration. So we're assuming that we have a neuron here that is surrounded by a solution of KCl, and we're asking what's going to happen. Because that neuron is more permeable to K plus ions, those K plus ions will move quickly into the axon down its concentration and electrical gradient to reestablish equilibrium. And the result will be to depolarize the axon and actually make it more likely to generate an action potential. Potential. Okay, and then for part B, what would happen if instead of adding more potassium to the outside, you add more sodium to the fluid bathing the neuron? And so our neuron, which I guess just looks like a worm, is bathing in NaCl. And I say NaCl just because I like I said in the previous, sodium also needs a counterbalancing ion, and so it also needs to be with something like chloride that matches its charge and is negative. So that's why we just are putting it in an NaCl bath. What's interesting about this, though, is we said previously this solubility works this way, where chloride is now more permeable than sodium. So what would happen is chloride rushes into axon, and this then hyperpolarizes the axon and makes it less likely to generate an action potential. Oh, yes, I totally can put part A back on the screen. Did not mean to scroll down that low. So I'm just going to summarize this one more time because I know this was a lot of information and it may have sounded daunting. In part A, we are thinking about the possibility of this neuron in potassium and chloride. And because potassium is rushing into the axon, it actually becomes more positive related to the outside, and we know that resting it is more negative. And because potassium is flowing down its electrical gradient, it depolarizes the axon and it makes it more likely to generate an action potential. In part B, it's the opposite, where because we're having sodium and chloride now on the outside, chloride is rushing in, making that axon more negative on the inside, closer to its resting, which means it's going to hyperpolarize the axon and make it less likely to generate an action potential. Oh. Cool. Nice. Okay. How is the nervous system organized? Neurons are single cells composed of processes called dendrites that carry information to the cell body and an axon that carries information away from the cell body. In simple terms, nerves are bundles of dendrites or axons or mixed axons and dendrites. Ganglia are connections of cell bodies, and brains are collections of ganglia. Processing and integration of the information occurs as a result of interaction within and between ganglia. So I know that this is a lot. I'm going to highlight to separate it for you. So dendrites give information to the cell body. Axon carries information away from the cell body. A for away, axon. Nerves are bundles of dendrites or axons or mixed axons and dendrites. And then finally, ganglia are collections of cell bodies, and brains are collections of ganglia. Yeah, this is a lot, but you're going to be awesome at it, so no sweat. Responding to stimuli requires, at a minimum, A, reception of a stimulus, which triggers B, an action potential in a sensory neuron, which synapses with C, the central nervous system, and generates an action potential, in D, a motor neuron, which causes a response, in E, a muscle or gland. In the space below, draw this minimum system. Be sure to name each neuron and label each of its parts. So I'm just going to go through this and write A through E. So reception of a stimulus, which we're seeing here. So this is A. We're seeing that happening on these dendrites. And that triggers an action potential in a sensory neuron. We're seeing that here. So this is B. Synapses with the central nervous system, which we see here. And this is the synapse, so we're going to write C here. And this generates an action potential in D, a motor neuron. This guy. And then which causes a response in a muscle or gland. Any questions so far? Okay. 2. The blink of an eye response is one type of simple reflex response that occurs. For example, in response to water splashing up out of the sink or a snowball aimed at your face. When this occurs, you blink, and only afterwards you realize why you blinked. What would you have to add to your diagram above to allow you to realize why you blinked? So for A, to add to the diagram, what I would add is an interneuron. Or another synapse to the sensory axon that led to an interneuron. So I'm just going to keep it at an interneuron. And that would carry the information to the brain, which allows you to realize what happened. And B, why does it take you longer to realize why you blinked than it takes for the blink reaction to occur? The simple reflex, basically, it just like cuts out the middleman. It goes right to the reflex. You don't have to think about it. So, simple reflex bypasses the interneuron. And so a positive outcome of this, not having to think about it, means that it can occur faster than it would if it would have the interneuron. And so most reflexes are designed, in our case, to protect the livelihood of the organism. And that's why they're so critical to have these reflexes. What composes the central nervous system, and how does it differ in general form and function from the peripheral nervous system? So the CNS is the integration and processing center. And it contains a lot of cell bodies and interneurons. Whereas the peripheral nervous system contain sensory neurons. And carries information from the sensors to the CNS and motor neurons which carry so sensory neurons, sensors to the CNS, and then they have motor neurons, which carry information from the CNS to the effectors, which just mean the things that do the thing. Awesome. Alright, the autonomic system is subdivided into the sympathetic and parasympathetic systems. How do the general functions of each of these divisions interact to maintain homeostasis? Sympathetic. So the sympathetic is known as fight or flight. It upregulates the glycogen to glucose conversion for energy. It secretes epinephrine. Increases heart rate. Increases blood pressure. And blood flow. To skeletal muscles and brain. So you can run fast and think fast. Alright, parasympathetic. Is antagonistic to sympathetic system. Promotes calming activities. Decreases heart rate and blood pressure. I'm just going to put BP. Increased digestion and glycogen production. And there's an exception to the antagonistic relationship between parasympathetic and sympathetic, where in the reproductive organs, the sympathetic and parasympathetic systems complement each other. Okay, moving on. Fibromyalgia is a condition that affects about 2% of the U.S. population. Its primary symptoms are chronic pain, difficulty sleeping, and fatigue that is not relieved following sleep. Other reported symptoms include jaw clenching at night, dryness of mouth and eyes, increased need to urinate, irritable bowels, muscle numbness, and tingling and headaches. A. Several researchers have proposed that fibromyalgia results from failed regulation of part of the autonomic nervous system. If this is true, failure of the regulation of which division of the autonomic nervous system could account for these symptoms and explain your reasoning. So, given the symptoms of pain, difficulty sleeping, constant fatigue, that just sounds like hyperactivity of the parasympathetic system. Just because we talked about how the parasympathetic is really for that rest and digest, the calming activities, the prolonged decreased heart rate, the tiredness, the difficulty sleeping and fatigued. The other things, such as the dryness of mouth and eyes, would probably be something else, as well as the muscle problems and headaches, just because that doesn't have to do with either fight or flight. Cool. To test their ideas, the researchers recorded the heart rates of both controls and individuals with fibromyalgia for 24-hour periods of normal activity. They observed that patients with fibromyalgia show greatly elevated heart rates both while active and when asleep. Does the addition of this information change or support the hypothesis you developed? So this would change because, as we said before, we thought that the trouble sleeping, the constant fatigue would result in a decreased heart rate, but knowing that they had greatly elevated rates both when active and asleep shows that it looks more like hyperactivity of the sympathetic system, which would elevate the heart rate and blood pressure and could lead to difficulty sleeping and chronic fatigue because you can't sleep. Let me just write this down before babbling off again. And also what I was going to add is that prolonged exposure to high levels of adrenaline that come with hyperactivity of the sympathetic system also leads to a reduced stress response. And so this may be the cause of the irritable bowel and bladder, as well as limb numbness, dryness of eyes and mouth. Okay. Oh, sorry. There's a loner down here. So then the final section we'll be covering is how the sarcomere structure affects muscle function. So number 1, how are sarcomeres arranged within muscle fibers and therefore within muscles? They are arranged linearly in myofibrils, and then myofibrils are arranged linearly in muscle fibers, which are organized into muscles. So I think in your textbook, a lot of times when they're discussing muscles, you'll see a picture of something like this, which has, you know, like it, that is the worst picture ever, but it's supposed to be a muscle where it's kind of breaking down into parts, and you'll see all these myofibrils into muscle fibers, which are all into one muscle. I'm just going to delete that, but I think you guys know what I'm talking about. So that's what it's asking for here. And then 2, describe the sliding filament model of muscle action. For example, what interactions power the movement of filaments in association with each other? Include trypomyosin complex, calcium binding sites, tropomyosin, myosin binding site, and actin in your description. So I find it best for this to kind of visually see it. There are great videos on YouTube and stuff, but I'll just do my best to draw it here. Sorry. So just looking at what a sarcomere is, we have just here. Oh, I could make a shape with that. Okay, I guess not, whatever. That's fine. So we have these things here. These are bands. And in blue, sorry. So in blue, we have actin. We have a filament in the middle. And it has these little things. These are myosin heads. No, I'm not drawing this well, but basically this is what it looks like in a relaxed conformation. We're seeing that it has, and if I were to more clearly depict it, these would be farther apart. But it has a clear zone here, and this is called the H zone. And that H zone is clearly defined in a relaxed conformation, but if it were to be in a contracted conformation, we would see things, specifically the actin, much closer together. Basically, they look like they're touching. So that's how the sliding filament model works at a microscopic level in this fashion, where we're seeing things getting pulled in closer together. And what it's asking for in terms of troponin complex, calcium binding sites, tropomyosin, myosin, troponin, sorry, just want to make sure. For this, we're getting even deeper into this actin filament right here. And basically, if we have actin that looks like this, they have these little sites. They kind of look like coconuts. And what these sites are is the myosin binding site. And it's usually blocked by what we call tropomyosin. And so this is what that tropomyosin complex is. So we call it tropomyosin because it's blocking the myosin binding site on the actin. And we have these other things kind of on the periphery, like here. And these are calcium binding sites. Now I know I'm throwing a lot of information at you, but basically what these calcium binding sites do is when there's a little bound calcium on it, it removes the tropomyosin and it makes it so these binding sites are now visible, and the myosin heads, these little guys, are able to reach up. I should draw this in pink. They're able to reach up and attach to the actin. And then ATP comes in, and basically what ATP does is it allows that myosin head to detach to actin, but not before it like slides this way. And that's how we get that pulling inward. I'll just draw this in completion. So then tropomyosin comes off. And then myosin. Well, myosin heads are able to bind and move that actin inward. And once again, there are a lot of videos on YouTube that are awesome, and those are computer generated drawings, so they're better. They're 3D, and they just show it. It's better that I would say this drawing is fine, but it's static, and with this sliding filament model, I think it's best and more beneficial to see it in action, because you'll see what's binding, what's moving, what's attaching, what's slide. Like, it's a lot of moving parts all at once. So just watching those videos is really helpful, and it was for me learning it. All right, 3. How do your answers to question 1 and 2, which are question to 1, how are they arranged? Describe the sliding filament model. How do they help explain why, at maximum concentrations, a muscle measures 70% of its extended, uncontracted length? As we saw, actin is pulled toward the center of the sarcomere. And just to clarify, this whole thing is a sarcomere. So anyway, actin is pulled toward the center of the sarcomere, and at maximum contraction, the sarcomere width is equal to the length of the thick filaments. And so with the relaxed sarcomere, the length of the thick filaments accounts for about 70% of the sarcomere width. And so that is why at maximum it measures 70% of its uncontracted length. A for 4, which is associated with a text in your textbook. It says acetylcholine, an excitatory neurotransmitter, is the neurotransmitter released into the neuromuscular junction. This release can trigger an action potential along the length of the muscle membrane. Describe the process involved. Motor neurons, synapses with a muscle fiber at a junction. Alright, action, potential. causes release of ACH, which is acetylcholine. This triggers an action potential in the muscle cell, which travels to the T-tubule system. Which releases calcium from the sarcoplasmic reticulum, which I'm just going to abbreviate as SR. And this contracts the sarcomere. That is supposed to be travels. Okay, just two more. I think I can squeeze them in. I'm going to put the Google form out one more time. If ACH always produces an excitatory response to the neuromuscular junction, how can we regulate which muscles in the arm, for example, are contracted and which are extended at any given time? For example, how can we bend the arm only partially? Muscles are arranged into motor units. The contraction of each motor unit is controlled by separate neurons. And so the degree of response depends on how many motor units have been stimulated to contract. And finally, C, severe calcium deficiency can lead to a reduction in bone mass. It can also have serious effects on the functioning of the nervous system and on the action of muscles. Explain what roles calcium plays in the activity of muscles. As talked about a little bit above, it is the release. So release of calcium from the sarcoplasmic reticulum that ultimately causes the muscles to contract. And so in cases of severe calcium deficiency, muscles can become weak or incapable of strong contractions because it is such a vital part of that contraction. It is the final step in what actually is the driving force for that muscle contraction. Whew. Awesome. So that is it for today. It is 3 o'clock. I put the Google Forms in a couple of times. Yeah, I hope that you guys have a good weekend, and I will see you next week.